what do promoters look like in bacteria, and how is RNA polymerase directed to the promoter. Okay. So, what I've already told you is that the promoter region is close to the transcription start site, technically includes the transcription start site. But the promoter, ideally, is only part of the coding strand. That's how you define a promoter. It's a part of the coding strand that RNA polymerase recognizes. Okay? So I've drawn my box around my promoter here, but I've only drawn it on the coding strand, not the template strand. Okay? What do bacterial promoters look like on average? Because they get wildly different from each other. Well, on average, bacterial promoters have a couple different features. So, if this is our +1 site, right? Our site of transcription, anything going this way we're going to call plus. So over here might be +400. Okay? So any place that we go behind the transcription start site, this is the minus direction. Okay? So when I break this promoter down and I make it a lot bigger, here's our +1 site. The very first base on the coding strand that will be made into an mRNA, right? 10 bases back from that, we have a thing called the -10 box. Why is it called a box? Well, initially when molecular biologists were learning these things, they noticed that if you deleted or mutated any of the bases inside this region, then that region didn't function. Okay? It didn't recruit RNA polymerase very well. So all of these are important, so they just called it a box. So we have a few bases between the -10 box and the +1 site, and we have a sequence of bases at the -10 box. This sequence is what we call a consensus sequence. What does that mean? Well, consensus sequences are sequences, so if we looked at 20 or 30 or 1,000 genes in E. coli, you might find TAT, in a 5' to 3' direction in 70% of those genes. Okay? That number might be a little bit high. It might be 50% of the genes. Sometimes one of the bases will be mutated, but generally you'll find TAT or something very, very close to that. So because you see that all over the genome in E. coli, right before +1 site, we say that's the consensus place where the RNA polymerase identifies a gene. Okay? But that's not enough, because we have 7 different sigma factors, and we have lots of different operons that need to be turned on at different times. So more than that, it's pretty much a combinatorial system, where not only do we have a -10 box, we have another box called the -35. The -35 box, much like the -10 box, has a consensus sequence, but this is extremely variable. So you might be asking yourself, do I want you to memorize the sequence of the boxes? No, I don't care. But you need to know that they're there. And the reason you need to know is because not all genes are on all the time. You need to be able to pick out that some genes are on at certain times, other genes are on at other times. Okay? So what's going to happen here? Well, this is our coding strand, so I'm just going to write a few bases into our coding strand. And that's plenty. Okay? Gene 1, remember, is somewhere up here. And there's a lot more stuff as we move in the plus direction. Okay? So, sigma factors recognize the boxes. So the very first thing that you'll get is sigma factors binding to both RNA pol and the DNA. So I'm going to draw the RNA pol complex with sigma factors here. And what's important here? What's important is that this entire complex, remember that's at least 6 proteins with at least one, or maybe I should say exactly one sigma factor, is directed to this area of the DNA. So now, this whole thing, I'll draw the RNA polymerase as a special Pac-Man looking molecule. What we've done is get RNA polymerase right close to the +1 site. Okay? But remember, DNA here is double-stranded. So RNA polymerase does not work on the coding strand. RNA polymerase writes an mRNA copy of the coding strand, its transcript, and it writes that copy from the template, if you recall. So what happens here, RNA polymerase actually starts using the bases from the template strand I forget miswrote my bases. Pardon me. RNA polymerase begins writing. I'm going to switch up colors here. I'm going to go to green for the new mRNA. And so the binding of RNA polymerase in this entire set of proteins opens the DNA up such that a bubble is formed, what we call the transcription bubble. And that's about 15 bases long. And the transcription bubble moves with RNA polymerase. So RNA polymerase starts moving down the template strand one base at a time, and as it reads the template strand, incoming nucleotides, in this case a U to bind with that T, comes up, hydrogen bonds with that A, so RNA polymerase poops out a U, then an A, or then another U, sorry, then it binds with this T to make an A, binds with the G to make a C, that G makes a C, this C makes a G, and so pretty soon our RNA polymerase, I'm going to erase it and move it down, there's our Pac-Man RNA polymerase, is moving down, leaving this mRNA, all of these orange bases, in its wake in the 5' to 3' direction, where every time it adds a new base, RNA polymerase is translocating down the template strand, reading the template strand in the 3' to 5' direction, and making a copy of the coding strand from the template strand. RNA polymerase will run all the way down until it hits the termination sequence. That is how transcription occurs in prokaryotes.